Nguyen. I'm your local rep. I actually live here in Gainesville for Olink Proteomics. I have been assisting researchers and clinicians and companies with multiplex products, whether it's at Olink or other companies, for over 13 years. So I'm excited to be a part of Olink. I've been here for about a year, and reintroducing all the communities and all the research communities in the Southeast to this product. And with us is Michael Forrester and Ann Lee. Michael is in Canada and Ann Lee is in Sweden. So we especially thank Ann Lee for presenting in the evening there. They're 7 hours ahead. And they're part of our scientific affairs group, which is a group in Olink that's responsible for essentially liaising and presenting with to the scientific community in various ways. They're a separate group from our application scientist group, like someone who would train you on an instrument. They're a separate group from that, and they're international, so they kind of travel around the world, working with scientists in their respective fields of expertise. So thanks for coming. Enjoy the food. I thought I brought too much, but I did not. And then Michael, I'll let you start.
Yeah. All right. So, yeah, I'm Michael Forster. I'm Scientific Director for Oncology and Rare Disease. And I have Andy Lind with me from Sweden, who's our scientific liaison for neuroscience. And what we will do in the next kind of like 45 minutes is trying to convince you that if you do genomics, you want to layer proteomics on top in order to fully take advantage of the samples and learn most about the patients that you're actually seeing. So just a second. So I would like to start out by actually quoting Sidney Brenner, who's developmental biologist extraordinaire and Nobel Prize laureate from 2002, who said that progress in science depends on new techniques, new discoveries, and new ideas. And tongue-in-cheekily added, probably in that order. And of course, I mean, we would like to convince you that now with the proximity extension assay, you have one of those new groundbreaking technologies widely available that do unlock or help unlocking the proteome of healthy individuals, diseased patients, learn more about complex phenotypes. So how does that map to our company's strategy or our company's vision? Is we do think that multi-omics is where it's at. So the integration of omics technologies is always going to improve understanding. You can layer on genomics, epigenomics, transcriptomics, cytomics, tissue phenomics, metabolomics, whatever omics technology you have. But we do also think that proteomics will always add more information, and on the margin for certain diseases will actually be a driver to really help the understanding of real-time human biology and the changes between health and disease. So where do we do what we do? Originally, we are a Swedish company, so we're based in Uppsala in just north of Stockholm. The IP that the entire Olink product portfolio is based on has been developed in Ulf Landegren's lab at the Uppsala Life Science Center. And we do have 2 large service hubs. So one of our service labs is in Europe, that's in Uppsala, and the other one is in Boston, meaning that our technology is basically available on a fee-for-service basis. You could send us your samples, we analyze them, give you back the data. However, as part of our externalization strategy, we've also partnered up with academic labs and with CROs that have either implemented the entire product portfolio or parts of the product portfolio, and then basically offer it to researchers in their vicinity. And we have a fair amount of service labs in the US available that could do the analysis for you. And we do not compete with our service labs in terms of price. So there is there is always that incentive. In terms of what is the not so secret sauce of the proximity extension assay, so what it is a dual antibody capture assay, meaning for anything to ever happen is you have to have two antibodies that bind to the same protein analyte, preferably in solution, at the same time. These antibodies are DNA tagged, so they have a short DNA molecule that is tagged onto them, and that will, of course, if the antibodies are in close enough proximity, enable complementary base pair binding, and that will subsequently enable a ligation reaction. And once you have a ligation reaction, you can imagine that you can read out small digital barcodes, either via NGS or via qPCR, and then make inferences about the concentration of the protein. That assay principle has a couple of advantages. So first, actually 5 advantages. The first one is specificity, because it's simple. 2 antibodies are better than 1, and 2 good antibodies against the same protein analyte will basically make cross-reactivity a thing of yesteryear. And then the second thing is sensitivity, because as soon as you have anything based on exponential readout, or any readout based on the exponential synthesis of nucleic acids, like either via NGS sequencing or qPCR, you have a more sensitive assay and you can cover a broader dynamic range of the proteome. You have the ability to multiplex. So right now, the maximum size of the protein library is 3,000 proteins. That is going to be extended, and then you also can reduce the assay volume. So you really only need a couple of microliters to read out of serum or plasma or other biospecimen to basically read out the entire library. And then lastly, since it is an immunoassay combined with a genomics readout, you can, of course, implement it along the entire value chain of pharmaceutical or biomarker development. So you can implement it in the discovery phase, but we also have a stab onto putting results from the discovery into the clinic. So with that, the product portfolio itself, it neatly bifurcates by assay readouts. On the left side, we have the NGS products. That's our discovery platform. It's called Explore. You can read out 3,000 proteins right now. You don't read out all 3,000 proteins in one reaction. You do that in increments of 384 proteins. So we have 8 blocks, and for every block, you need roughly 1.5 microliters, or something like 12 to 13 microliters of serum or plasma will enable you to read out 3,000 proteins. And then on the right side, you have the qPCR readout. In the middle, that's our midplex portfolio. So that's the target 48 or the target 96 plex. So these are you read out roughly 48 cytokines or 90 proteins that are thematically and spotted together, and then you can look at proteins that are specifically for inflammation, for oncology, cardiovascular, and neurology, things like that. You can do this in your own lab because we have a development of our own instruments. So we have a qPCR machine available that's called Signature Q100 that allows for the readout of anything with qPCR. And then on the right side, this is a product that's going to come out hopefully by the end of the year. So that's called Focus. And what Focus will enable you to do is basically take your favorite biomarkers up to 21 or 23 of them, spot them onto your own test, combine them in one test, and then potentially validate it in tens of thousands of patients as an LDT. So you could theoretically design a project where you use the Explore platform for discovery, and then you use the Focus platform for clinical implementation. It is somewhat hard to make this available for the entire platform because that entails a lot of validation, but that's what we do up front. So there it is going to be a stacked release. Not all proteins will be available by the end of the year, but we're striving to have all proteins, the entire library, available. In terms of what the library entails itself, so it's 3,000 proteins, 1,400 of those are new biomarkers, and 900, so roughly a third, is Olink specific antibodies. So that is antibodies we developed ourselves. We do have customer wish markers as well. So any future extension, we always go into the scientific community and we would like to know what are your favorite targets that we currently not cover. The entire library is online. You can check by UniProt numbers, is my favorite protein included? And if not, you can shoot us an email, and if there is a good rationale to add this protein and we can make the antibodies, we will add it for future iterations of the library. And by the end of the year, the library is going to have available 4,500 targets. So we are going to release another 1,500. What is covered? The library is tilted or tailored towards the circulating proteins. So proteome, so 800 biomarkers are somewhat exploratory, but they're all annotated to be detectable in blood. 450 are going to be the human secretome. We do have a large focus on inflammatory proteins. So we have up to 1,200 proteins that in one way or the other relate to inflammation, and we do cover 600 known drug targets as well. So if you're interested in looking at especially mechanism of action studies, you can do that even with the discovery library. So we also have 60 high abundant proteins included. And why would you do that? So those high abundant proteins are typically proteins that you can pick up by mass spectrometry, and that kind of indicates that our platform is complementary to mass spectrometry. And I'll show you an example of that in a minute. In terms of orthogonal validation, what people have done in terms of independent labs have taken us to task and have compared us, especially like the Midplex portfolio, against the Plexin platforms like Luminex or MSD. And what they've basically found is we have better sensitivity, simply because we have the qPCR readout. We have a wider dynamic range. So even if you assess cytokines, you can use us for rare cytokines that you can't use Luminex or MSD for. We have better linearity and in general, the highest quality of data. And then on the proteomics side, so on the classical proteomics, on the mass spectrometry side, when people use this here to interrogate the circulating proteome, you do find something between like 300 or 500 proteins. It's often the classical plasma proteins, the albumins, the HDLs, the LDLs. And you have a couple of leakage proteins that do have value as biomarkers, like PSA or troponin, things like that. So for that, we have our 60 targets. And then, of course, since we do have this NGS readout, we cover a broad dynamic range. So whereas you will kind of like end in somewhat of the microgram range for proteomics, you can actually go into the femtogram range using the Olink Proteomics library, while at the same time always compare your data set you've possibly derived with mass spectrometry through these high abundant proteins that we also have included. So in terms of sample types, so as I already mentioned, largely or the initial validation for us is always serum or plasma. So the library is geared towards proteins that you'll find in serum or plasma as well. Not all targets, but a large part of it. But that doesn't mean that you can use us on other biospecimen or other matrices. We actually have been used in like roughly 24, anything from urine to saliva, tissue lysates, cell culture media, aqueous humor, tear fluid, pretty much any bodily fluid you can imagine. Also, CSF is a large part, especially for the neurology studies. The only two things that don't work is formalin fixed tissue. So if you the formalin fixation will interfere too much with the binding epitopes of the antibody, so that will not guarantee a good readout. And then the second thing is actually fecal matter. So people have tried to use us there, but unfortunately the large polysaccharide molecules interfere as well with the antibodies, and then you won't have a good readout. So those are the a priori two biospecimen we always say don't use us on that. And then the other thing is you can actually, if you plan to use the Olink library on a new type of biospecimen or a new matrix, you can shoot us an email. Because our technical services and our R&D already have oftentimes established protocols available that will help with the design of your experiment. So we are proven leader. We work with 700 pharma companies. All top 20 pharma works with us. We have something between 3.5 million samples analyzed in our own service labs. We are approaching 1,000 publications that actually use Olink in one way or the other. We work with a lot of KOLs, and we do have 3 consortia that basically specifically use Olink for answering questions either within like complex trait genetics or proteogenomics. That's the Scallop Consortium, the Coral Consortium that's specifically for neurodegenerative diseases, and the Colibri Consortium that's specifically for IBD. So now along this line of implementing Olink alongside the pharmaceutical value chain, we are present in terms of like, if you want to new drug discoveries, large population or cohorts use Olink. I'll present some examples in a minute. We're also used in preclinical research, so cellular models, somewhat in organoids, because the library and the small sample input you need lends itself towards that. Clinical development, we are part of clinical trials. So right now there are 42 clinical trials along different diseases that stratify patients using Olink Proteomics PIA assay. And then lastly, this regulatory approval, that's pretty much the focus platform. So that's up and coming in terms of you may not be able to build an in vitro diagnostic test because right now it's a research use only application, but you can at least offer an LDT. And we have an example on that as well. In terms of looking at discovery projects, there can't be an Olink talk without talking about the Pharma Proteomics project. So the Pharma Proteomics project is in collaboration with the UK Biobank. It's 12 companies that came together to work with the largest biobank in the world that contains 550,000 individuals, and roughly 10% of those were to be tested or were tested with Olink's Explore 1.5 K discovery platform. And the results are actually released as a draft manuscript right now. So at the beginning of the summer, Benjamin Sun and under the supervision of Chris Whelan, they published the initial manuscript. They tested 54,000 participants, and their study design was as follows. So they had the 54,000 participants. They said we do roughly 1,500 proteins. They tested the intra-sample variation on samples they continuously reran to basically check for batch effects. They didn't find any of those, and the mean intra-sample variation was 6.2%. Their genotyping was 22 million SNPs, so it wasn't super tight, but relatively tight. And then their cohort was split into 3 different subcohorts. So first, they took random samples, roughly 46,000. That was just to basically mirror the UK population. Then they had roughly 6,500 individuals that they were having certain diseases, and they were enriched for 120 different diseases. They were a bit different from the populational cohort. They differed. They had a higher BMI. They were older. They had a higher smoking prevalence, and they were somewhat more multi-ethnic, while also having a smaller proportion of women. And then they had a COVID cohort. So they had roughly 1,300 individuals that were participants of a serial COVID imaging study. They're also different somewhat from the general populational cohort, but these, so they did have an aspect of COVID, and actually the largest proteogenomic analysis of COVID-19 within that study. They split it for bioinformatics analysis into, I can't exactly see it, I think it's 36,000 individuals that were white Englanders, so that was their discovery cohort, and then everybody who self-reportedly was more multi-ethnic, that was their validation cohort. And they still, even though the genetics of those two cohorts is somewhat different, they could validate more than 90% of all the initial pQTLs they found. In terms of results, I won't go into the details of that paper because it's extremely long, just picked some illustrative examples. One of it is that with these populational cohort study, there is now the advent of system biology and proteogenomics. They looked specifically, Benjamin Sun and colleagues specifically looked at circulating proteins. It's always easy to analyze. And then secondly, there are very, very common drug targets. So they looked at cytokines and complement. And then they looked at what type of pQTLs they found. They did find the example here they used is the IL-15 pathway. They found a cis-pQTL for IL-15, so basically a genomic variant that maps to the IL-15 locus. But they also found the receptors, the alpha chain of the IL-15 receptor, the beta chain of the IL-2 and the IL-15 receptor, and the adjacent kinases JAK1 and JAK3 with trans pQTLs that all, in one way or the other, change IL-15 levels in circulation. And then they did a modeling study asking what is kind of like the ideal population size. So if you want to identify cis-pQTLs, meaning new drug targets, they kind of found that between 5 and 10,000 individuals analyzed, there is a plateau phase of cis pQTLs, meaning that's kind of like the ideal or the minimum study size you should consider. But for trans pQTLs, so if you really want to do pathway mapping, they showed that for the IL-15 pathway, they had to use the entire 54,000 individuals to find all the trans pQTLs. There is no plateauing, and that means basically additional individuals will on the margin always add additional valuable information. And that has to some extent implications for therapy, because cytokines are often drug targets. I just picked tofacitinib. Xelian's is the JAK1, JAK3 inhibitor for chronic inflammatory diseases. So if you wanted to study that in a pharmacogenomics type of background, you could actually use this type of study design as well. And then the other example, I mean, just for actuality's sake, is actually proteogenomics of COVID-19. They did the largest proteogenomics study with 1,300 individuals. They then were interested, okay, what can we, they chose as clinical readout largely COVID complications assessed by ICU visits. And then they kind of asked, okay, what are the circulating proteins that govern that? And they looked at lung-specific proteins. They actually found that mucin 5B, a SNP in the mucin 5B promoter controlled mesothelin, LAMP3, and the surfactant protein D in trans. And that protein module was associated with COVID-19 hospitalization, and that SNP was actually mapping strongly to COVID hospitalizations and to basically COVID complications. And then they did the same analysis. For immune-specific genes, and there they found the ICAM gene cluster on, I think, chromosome 19. They also found that signal overlapped with a missense variant in TYK2 and a pQTL in the IL-12 receptor beta subunit. It kind of points towards an immune protein network that controls the hyperinflammatory reaction that's associated with COVID complications. It makes sense because ICAM genes point towards immune cell extravasation, whereas TYK2 and the IL-12 receptor subunit, they point towards like either dysregulation and interferon signaling or some like pro-inflammatory immune phenotype. So in general, as a proof of concept, this shows that proteogenomics, that you can actually identify potential pathways with which you could interfere in order to help patients that suffer from severe COVID and from severe COVID complications. So that these are the key takeaways in terms of systems biology. You can now study pathways in these large populational cohorts. You don't have to go for single targets, and that of course has reverberations for drug development. And then secondly, on the infection biology side, you can use these populational studies to not only identify lung-specific, so organ-specific signatures that in one way or the other predict pathology, but also immune-specific signatures. So with that, I would like to also lose some words onto the SCALLOP consortium. So the SCALLOP consortium has somewhat of an anachronistic name. It's systematic and combined analysis of Olink proteins. So that's basically researchers from different parts of the world, mostly from Northern Europe and from the US, coming together, bringing together cohorts. So it's 33 researchers and 48 cohorts and roughly 65 to 70,000 individuals with mapped out for different phenotypes. under the auspices of Anders Malostik, who is the scalloped chair, who plies his trade as a professor at Karolinska Institute. And that's basically our initiative that targets similar things as the UK Biobank. So basically looking at disease phenotypes, trying to identify meaningful genome, protein, phenome triads that can be that can be targeted pharmacologically, and they also have a large project on drug repositioning. One of their initial publications was actually 1.5 years ago in Nature Metabolism. They just took the 190 protein S plate, so one target 96 assay. They investigated 31,000 individuals, and they found 485 pQTLs using the Mendelian randomization method. They also found that they found 25 causal relationships, and of which the majority, 14, were novel. And they actually also predicted some phenotypes that for known drug targets, like for example, toxicilumab or anakinra, targeting either the IL-6 pathway or the IL-1 pathway, established for chronic inflammatory diseases, that there is an overlap with certain phenotypes that are associated with cardiovascular disease. So with that, on the preclinical research side, so going a little bit more into like cellular models, we do have one study that Stefan Nelander's group from Uppsala, who basically used Olink as part of their drug screening study in glioblastoma. What did they do in this study? They took patient-derived cells, cell lines, roughly 100 cell lines. They tested roughly 1,500 drugs, and they had this multi-omic approach where they layered genomic information, transcriptomic information, and proteomic information in order to learn, okay, what are the active drug classes that could be used in these glioblastoma patients? Can we identify response predictors? And can we find meaningful combinations that in one way or the other overcome resistance? What they used is they also used the Target 96 panel. And the rationale was basically that we had the right targets. They used the cell regulation and the development panel. And what they found is they found a nice need bifurcation. So basically the glioblastoma cultures could be separated into one of two groups. Either they were proteasome inhibitor sensitive or they were proteasome inhibitor resistant, and that overlapped with a genomic signal. So you basically had mutually exclusive mutations either in the P53 locus or in the cyclin 2A to B locus, and that overlapped with this pharmacological phenotype. They confirmed the entirety in cell and xenotransplantation models. So they basically said, hey, if we have these cell lines, can we pick out cell lines for which we would predict they're either sensitive or resistant? They did the prediction model, or they incorporated the proteomics data for that prediction model. And then they put these into animals, and they found that the cell lines they behaved exactly as they predicted from their previous data. And then lastly, they also found that you should use BCL2 family inhibitors or P53 activators in combination to overcome resistance for bortezomib, for proteasome inhibitors. So the key takeaways from this study, multi-omics, including the proteomics piece and this pathway analysis, actually improves drug response, because without the proteomics piece, they wouldn't have found these potentiators that could overcome resistance. And then you can actually predict drug responses and have opportunities for drug repurposing, which is, of course, for glioblastoma, incredibly interesting because it's super hard to treat and it's incredibly deadly as a cancer. So then in terms of clinical development, that's just for actuality's sake. We have been used within the field of CAR T-cell responses as well. So a company used us in an early phase clinical trial to look at hematological malignancies using a CD19 targeting CAR T-cell therapy. Paper was published in 2018. They either had B-cell lymphoma patients or they had acute lymphoblastic leukemia patients. What they looked at is they looked at CAR T-cell persistence. So they did that with qPCR readouts, and then they tried to predict therapy response, including with Olink's Target 96 immuno-oncology assays. And what they found is basically, I mean, they had a couple of 6 out of 15 patients in that trial had complete responses. 3 patients were still in complete response after 3 months. So it wasn't like, I mean, it was a late stage hem-onc trial, so it wasn't incredibly great on the response side. But they found that if you have low levels of myeloid-derived suppressor cells and low levels of certain soluble immune checkpoints that were included in the Olink library, that will in one way or the other at least correlate with the response rate. But the best predictor were soluble factors like IL-12 or LAMP3 or TRAIL that argue for good pro-inflammatory immune status. So if you have a good pro-inflammatory immune status, at least according to using these readouts, you will have a better, or you can actually reasonably well predict response. So that shows that Olink's targeted proteomics platform, even if you just use mid-plex assays, is a suitable tool for the identification of predictive biomarkers, even in a kind of like numbers constrained setting of a CAR T-cell trial. And the authors in the paper, they argue that that has implications for patient stratification or trial design, because potentially you could stratify patient a priori before enrolling them into a CAR T-cell trial. And with that, I'm actually at the end of my part of the presentation. I will hand over to Anli for the neurology case studies.
Thank you, Michael. That was really interesting, also for me. Just gonna check that I can switch on my video here. So there, can you see me now?
Yes, all good.
Great. So I had mapped around 15 minutes for this, but I want to make sure we have time for questions. But let's get started. Ariel, do you have another opinion?
No, I think that's fine. I think if it goes over with questions, people who want to stay for that part are welcome to.
Great. Okay, so I'll just dive in. So I'm delighted to spend the next 15 minutes sharing with you 3 studies that have used Olink to investigate different aspects of neurological diseases. First, we'll look at a study going from the left to the right in this slide, investigating different aspects of neurological diseases. Investigating disease signatures and models to classify AD. And then a second case is a set of studies that investigate potential biomarkers for triage and outcome prediction in traumatic brain injury. And then we will round off with a study focused on developing a laboratory developed test, an LDT, for multiple sclerosis disease activity testing. So let's have a closer look at this first neuro study. As most of you know, Alzheimer's disease is the most common form of dementia. It affects around 30 million people worldwide and causes suffering and disability to patients and families. It's a high-cost disease, and with aging populations worldwide, progress in AD research is obviously a matter of relevance to society at large. And much effort has obviously been put into the many AD treatments that are in clinical trials and some on the market, but plenty of work remains. And AD pathophysiology, being complex, as I would say nearly all diseases, and associated with A beta and tau pathology, inflammation, and synaptic injury, further mechanistic insights can boost development of effective AD treatments. Additionally, as was a big topic of discussion during the last AAIC meeting in San Diego in August, 2 months ago, where I believe some of you might have attended, we need less invasive blood-based tests for AD screening to take advantage of the preclinical time window of around 8 to 12 years or even more before symptoms arise. And this is an area of unmet potential under intense development. Most of the markers under consideration are what we would call the usual suspects. And in this study, the group of Nancy Ip and the Hong Kong Institute of Science and Technology, together with researchers at UCL, UK, aimed to look outside the box to assess if there were previously unidentified AD-associated plasma proteins that were suitable for a blood-based test for AD screening and staging. And here on the top right, you can see a schematic of their 2 independent cohorts of well-characterized AD patients and cognitively unimpaired control subjects. There was a discovery cohort and a validation cohort. They chose to run 13 Olink T96 panels. I believe at the time that was the largest you could go because there was no Explore product at that time. And that made this the most comprehensive publicly available profiling of the Alzheimer's disease plasma proteome when it was published last year. In the volcano plot on the bottom left, you see associations among the 1,160 analyzed plasma proteins. The blue and red dots indicate proteins in patients with AD that were either down or upregulated compared to controls respectively. And the resulting 429 AD-associated proteins were then subjected to correlation analysis, identifying 19 hub proteins listed in the table on the bottom right. These 19 proteins were incorporated into a model to classify clinical AD, and that model was cross-validated in their validation cohort. In this graph, you see the ROC curves that represent the performance of 3 different AD classification models in the discovery cohort. So the ROC curves of the models based on classical plasma ATN biomarkers, that is A beta 42/40 ratio, tau, and NFL levels, is represented by the yellow line. The novel 19 protein biomarker discovered in this paper is represented by the blue line, and the red line represents performance of the combined model. The embedded bar chart in the top right corner of the graph shows that the area under the curves, according to the 3 classification models, were 0.87 for the classic markers, 0.98 for the novel markers, and 0.99 for the combined model. So the new model outperformed classic markers in both cohorts. This is just the discovery cohort, but it was a similar result in the validation cohort. So this study established a high-performance plasma biomarker panel for AD in this particular population. The panel can serve as a critical foundation for developing a blood-based test for AD screening and staging. There's also staging data in this paper that is really interesting, but I couldn't include it for the interest of time. The data offer ample novel candidates for drug development and add a layer of understanding to AD disease pathophysiology. So let's move on to the second case, which is a series of studies that are focused on traumatic brain injury. And it starts with this concussion study conducted at Western University in Ontario, Canada. So sports concussions can be difficult to diagnose. Misdiagnoses can expose athletes to greater risk of injury and long-term neurological disability. Objective and easily accessible biomarkers to aid concussion diagnosis is therefore a much needed step to protecting youth brain health. To contribute novel, potentially useful blood-based biomarkers, these investigators designed a study based on subjects detailed in the table. The subjects were enrolled in their academic sport medicine concussion clinic. And the first column, you see their clinical characteristics of the 11 enrolled adolescent athletes that were suffering from sports concussion. In the middle column, the characteristics of their 24 sex, age, and activity matched healthy controls. And clinical evaluation and blood draw in the study was within 72 hours of injury. Samples were then analyzed with Olink Explore 1536. And here are their main findings. Out of the 1,472 assayed plasma proteins, 6 were considered significant between the cohorts. In the left picture, you see a box plots comparing plasma expression of these 6 proteins in concussion patients and matched healthy controls. 5 proteins decreased and 1 was increased. These proteins have known roles in modulating tissue inflammation and regulating integrity of cerebral microvasculature. The second figure on the right contains the ROC curve analyses of several protein combinations for concussion diagnoses. And when predicted values for the 3 leading proteins, as determined by regression analyses, were combined, the area under the curve increased to 0.98. So taken together, these proteins are interesting targets for potential point-of-care tools for screening adolescents for concussion. And obviously, validation is required in much larger cohort. So finally, on the TBI topic, I just want to highlight 2 recent TBI publications that unfortunately are not open access, and I didn't pay for a license to share their results here. But if you're interested in this topic, I highly encourage you to look them up afterwards. But I want to mention some of the available information. So they produce very interesting results pertaining to individualized patient care, which is essential to reducing global burden of TBI. If you're interested, please have a look. But on the left, you see another the publication from the same group that published the concussion study I just shared. But here they focused on markers for outcome prediction in severe TBI. So they found 2 biomarker pairs that were obtained shortly after ICU admission that could accurately predict outcome. Specifically, when they combined von Willebrand factor with either WIF1 or CSF1, it resulted in excellent mortality prediction with an area under the curve of 1.000 for both combinations. And these 2 protein biomarkers combinations greatly outperformed mortality prediction of 2 commonly used brain injury biomarkers, NSE and S100 beta. On the right, you can see a publication from the prospective observational study called CREACTIVE, which stands for Collaborative Research on Acute Traumatic Brain Injury in Intensive Care Medicine in Europe, where concentrations of 107 biomarkers, including an Olink T96 inflammation panel, were quantified in blood samples collected from 80 TBI patients on ICU admission. 6 biomarkers were associated with indicators of primary or secondary brain injury. 3 of them were glial proteins that are well recognized, glial cell-derived neurotrophic factor, glial fibrillary, GFAP, and S100 beta as well. And then 3 cytokines, stem cell factor, and 2 fibroblast growth factors, 23 and 19. And when subjects were grouped into 3 clusters according to their admission levels of these 6 proteins, the prognostic accuracy of established clinical parameters as well as computed tomography were improved. So these results warrant validation in larger independent cohorts, and I believe that work is ongoing. Nonetheless, the results are a very promising step towards personalized medicine for TBI patients. So in this third and last case study, it is from Octave Bioscience, which is a biotech company working to improve multiple sclerosis clinical care. This slide just shows a snapshot of their research poster presented at the annual ECTRIMS meeting in October 2021. So as represented on the left, multiple sclerosis is a progressive, lifelong debilitating disease affecting 2.8 million people globally, nearly 1 million in the US alone. The disease burden leads to 50% of patients being on disability within 12 years. Multiple sclerosis being heterogeneous, it also has a complex pathology driven by inflammation and demyelination. And current disease assessment tools are subjective and quantitative. So MS is a high-cost disease, and Octave's contribution to this, to improving the situation, is providing a care platform, which includes blood-based protein measurements of MS disease activity. So they aim to provide healthcare professionals with a tool to detect biological warning signals of relapse and silent disease progression in people with MS really early, ideally before ideally before symptoms arise. To this end, they needed a very sensitive, multiplex, high throughput protein biomarker tool, and they partnered with Olink for both the research phase and the manufacture of a custom Olink focus panels for development and validation at Octave. The flow diagram on the right summarizes Octave's 4 steps of research and development. First, Octave conducted a feasibility study. Second, they defined an MS proteomics signature by performing deep scans of small volumes of blood samples from 305 deeply phenotyped MS patients. Then in the third phase, in an iterative process using statistical, analytical, and biological factors, 21 proteins were selected for inclusion in the custom absolute quantification panel, which was manufactured by Olink. And the 21-plex panel was then developed thoroughly analytically validated at Octave. And in the last phase, Octave validated their assay clinically in more than 600 samples from independent cohorts from 4 different sites. So starting with a candidate biomarker pool of over 1,400 proteins, narrowing it down to the 21-plex, and now Octave have an 18-plex panel laboratory developed test for MS disease activity that is going through additional testing. So Octave's new panel outperforms neurofilament light for all 3 disease activity and clinical radiographic endpoints that are tested. The assay provides real-time insight into the activity of 4 main biological pathways involved in multiple sclerosis pathophysiology. That's immune modulation, neuroinflammation, myelin biology, and neuronal integrity. And with this new tool in their hands, they're now moving forward to next steps. So here on the right, you see a selection of their most updated scientific posters from this year. So I summarized the highlights on the left. So they tested the panel in a real-world clinical practice population of people with multiple sclerosis, and the LDT performed reliably against key endpoints. Also, 6 of the panel proteins were correlated with relapse in a longitudinal sample set from a new cohort of 69 patients, and then they analyzed 6 MS treatment classes, and 17 markers were actually associated with treatment, indicating that the test has a potential to capture real-time treatment impact on patient biology. So in summary, I've shared from the neurology fields 3 cases that have produced interesting results. A 19-protein AD classification model outperforming classic ATN markers, providing novel markers for AD staging and future drug development. Several interesting candidates for TBI triage biomarkers, as well as ICU prognostic markers in severe TBI, and an LDT for multiple sclerosis disease assessment that's undergoing validation in real-world clinical practice. So, however, as most experienced biomarker investigators know, it takes a village to raise a real-world useful biomarker, and plenty of work remains. So Olink has heard from our customers and our community the need for more collaboration, and that is why we sponsored collaborative initiatives in several fields that Michael has already mentioned. So these are the 3 consortia. I won't go into details on that now. I could speak more to anyone who's interested specifically in the Coral Consortium at a later time point, but I want to finish there since we have only a few minutes left. Thank you.
Does anyone have any questions? Oh,
Oh, sure. So the question concerning the difference between Olink versus Soma. So, I mean, the first issue would be in terms of like, what's the specific